Understanding the GARD assay, a full introduction to the technology platform. GARD stands for Genomic Allergen Rapid Detection. The development of all GARD assays is based on the same principle, which includes four key elements. The first element is use of a relevant biological cell system. The second element is a training data set to identify endpoint-specific genes that can be used as biomarkers. These identified genes make the third element, a so-called genomic biomarker signature. Finally, the fourth element is a prediction model used to classify unknown substances. We'll go through each of these one by one. The first step for constructing a mechanistically relevant in vitro assay is to select an appropriate biological model which can reflect the adverse toxicological endpoint we are trying to predict. By looking at the adverse outcome pathway for sensitization, we see that the dendritic cells are highly relevant in this pathway since they are responsible for driving the polarization of naive T cells into either a Th1 or a Th2 response that is associated with the development of skin or respiratory sensitization, respectively. Therefore, in the GARD assays, we use a human dendritic-like cell line called the sensicell cell line. This is a monocytic cell line that has been differentiated to acquire many characteristics identical to those of primary dendritic cells. Like many primary cells, the sensicell cell line can take up and present antigens on MHC classes 1 and 2, as well as on CD1D. They respond to pro-inflammatory cytokines by upregulation of co-stimulatory molecules such as CD80 and CD86. And importantly, they contain the functional elements involved in regulation of Th1 and Th2 polarization. Having defined the cellular system and knowing that the cells react differently upon encountering antigens, the question is then how we monitor the response of these cells to sensitizers. First, they are equipped with various receptors that recognize foreign substances. These receptors may in turn activate various self-defense mechanisms of the host, such as cellular stress responses like oxidative stress. Finally, they upregulate receptors that allow them to migrate and communicate with other cells of the immune system. How then can we monitor all of these events at the same time? Since all of these changes in the cell are carried out by proteins, controlled by the transcription of DNA, we believe that genomics, or more specifically gene expression analysis, is a very efficient way to get a complete view of what is happening in a cell upon chemical stimulation. With this in mind, we hypothesize that there should exist genes in these cells that are differentially regulated depending upon the stimuli. If we can identify such genes, they could be used as predictive tools. That leads us to the second element, the defining of a training data set. A training data set comprises a set of information from a set of well-characterized chemical substances with known outcomes. This means that the chemicals should be defined according to their sensitizing capacity and ideally based on human data. Moreover, the reference chemicals should cover a wide variety of chemical reactivity domains and at the same time include different industrial applications. This can help capture the different modes of action and develop a model with a large applicability domain. To identify genes that are differentially activated in the sensicell cell line, we expose the cells to the reference substances for cellular stimulations. After 24 hours, we extract the RNA and measure the complete transcriptomes of the cells using the Affymetrix microarray platform. For every reference chemical in the training set, we measured the whole genome, which is comprised of 29,000 genes. This leads us to the next technology element, the genomic biomarker signature, which was identified by observing how the substances in the training data set affect the sensicell cell line. Of the 29,000 genes measured, we narrowed it down to a subset of genes that we identified as being differentially regulated depending on whether the cells were stimulated by a sensitizer or a non-sensitizer. In contrast to the traditional approach where one often measures a single biomarker based on previous experience, this holistic approach is completely data-driven and unbiased. It provides us the opportunity to see the bigger picture of how the cells react to different chemicals, and it identifies novel predictive biomarkers for sensitization. Using the GARD skin assay as an example, we eventually mapped out 200 genes that were differentially regulated depending on whether the cells were stimulated with sensitizers or non-sensitizers from the training set. These 200 genes are the so-called biomarker signature of GARD skin. Looking deeper at these 200 genes, we found that many of them are known to be involved in a broad spectrum of processes for the development of the skin sensitization endpoint. When we map the genes to the various key events in the adverse outcome pathway, we find that they are involved in multiple key events required for the endpoint represented by that AOP. We believe that this is why GARD skin has such a high predictive accuracy, because it not only measures one specific biomarker, but measures a whole set of different biomarkers that are immunologically relevant to the whole sensitization endpoint. Once we identified our biomarker signature, we wanted to transfer the gene expression measurement from measuring the complete transcriptome of the cells to a higher throughput, more streamlined platform that measures only the genes relevant to the signature. The Affymetrix platform used for identifying the biomarker signature is extremely useful when needing to observe the whole transcriptome of the cells. But the platform does not fit very well with routine testing as it is very labor-intensive with low sample throughput. Therefore, we switched to another hybridization-based technology platform called NanoString, so that we can perform the analysis in 22 hours with only 15 minutes hands-on time. We also simultaneously increased the sample throughput to as much as 384 samples per day. What you see here in this figure is a comparison between the Affymetrix microarray data on the right and the novel NanoString data on the left. As you can see, the gene expression measurements were reproduced and validated on the NanoString platform, demonstrating that the genomic biomarker signature is a stable and fixed biological endpoint, independent of technological means used for gene expression measurements. All GARD assays are based on the same technology platform, using the same biological system of the SensaCell cell line and the same prediction model. The differences between the assays are the training data that was used and the identified genomic biomarker signatures. The signatures for each consist of 200 genes for GARD skin, 51 genes for GARD potency, and 28 genes for GARD air. GARD skin medical device and GARD skin dose response utilize the same 200 genes as the GARD skin assay. After identification of the predictive biomarker signature, we need to define the prediction model that uses the gene expression data to classify unknown test substances. Developing the prediction model is a process of teaching the computer to recognize sensitizers and non-sensitizers based on their capacity to induce different gene expression profiles. By doing this, the computer can make a prediction by comparing the gene expression pattern of the test substance against existing patterns of known sensitizers and non-sensitizers from the training dataset. To assist in this process, we use a technology of pattern recognition, often referred to as machine learning. In the same way that people learn from their past experiences, a computer can be trained to learn from a set of data. This is the concept of machine learning. Here is more on how machine learning works and how we use this powerful technology in the GARD assays. The machine learning algorithm learns by observing a set of objects with known expected outcomes. If we consider our training data, we have the gene expression profiles from a set of well-characterized compounds that are either sensitizers or non-sensitizers. Using this training data as input, the machine learning algorithm learns to distinguish expression profiles induced by sensitizers and non-sensitizers. To classify an unknown sample, the expression profile induced by the test substance is compared to the expression profiles of the training samples and classified as either a sensitizer or a non-sensitizer. In this example, the expression profile of the test substance is most similar to the expression profiles of non-sensitizers. It is therefore classified as a non-sensitizer. This figure is a graphical representation, but we can also explain the process in a mathematical fashion. Mathematically, the information in the heat map can be expressed as a gene expression data matrix containing the information of the class label, sensitizing or non-sensitizing. In the machine learning algorithm, this now becomes an optimization problem. What the machine does here is to assign values to the different genes with the purpose of optimizing the discrimination between the sensitizers and the non-sensitizers. This leads to a fixed mathematical algorithm. The gene expression data from an unknown test compound can then be input into the fixed formula, and the model provides a prediction for the test compound. Having discussed how the technology is developed and how it works, I also want to describe how the GARD assays are actually performed in the lab using the GARD skin assay as an example. In step 1, the Senza cells are exposed to the test substance for 24 hours at the highest concentration where 90% of the cells are viable, or at a maximum of 500 micromolar. For step 2, the RNA is extracted and is used to measure the gene expression levels of the biomarkers using nanostring technology. Lastly, in step 3, the gene expression pattern is analyzed by the machine learning algorithm in the GARD data analysis application, providing a binary answer, sensitizing or non-sensitizing. No expert judgment is needed.